Human cytomegalovirus, HCMV, is a herpes virus that causes lifelong infection in people who are infected. While most who are infected are asymptomatic, it's often very dangerous for immunocompromised people. Within HCMV's genome, there is the protein UL56, which functions to cleave the viral DNA after it has been replicated, which means that new viruses can be made. Mutations in the UL56 are studied to see how resistant it becomes to antiviral drugs. The mutation I studied was E237D. This means that the amino acid of position 237 of UL56 has changed from its original glutamic acid, E, to aspartic acid, D. Both are polar acids that are negatively charged at physiological pH, but aspartic acid is smaller than glutamic acid. To create the mutated gene, first, a bacterial plasmid, PND, was cut into a linear plasmid with restriction enzymes NOT1 and XBA1. Then forward and reverse primers were created that contained the E237D mutation with PCR. These primers also contain the mCherry gene. This is a red fluorescence which helps to track the location of the mutation. The primers and the PND plasmid were added to a recombinase enzyme to allow recombination. This resulted in a circular plasmid with the mutated UL56 E237D mCherry. This mutated plasmid was transfected into E. coli cells and incubated. Transfection does not mean that the plasmid is integrated into the E. coli's genome. It exists separately. The cells transfected improperly would not have the antibiotic resistance from the mutation, so they were incubated on plates containing ampicillin. This weeded out any of the improperly transfected cells. A second plasmid, called a reporter plasmid, was also transfected into the cell. This is to actually answer the question of what effect the mutated UL56 has. This reporter plasmid has a promoter, an A site, and a CFP gene. CFP fluoresces blue. If there is UL56, it would bind to the A site of the reporter plasmid, which means that the CFP gene would be blocked, which means less blue fluorescence. This means that if the UL56 was alone, it would only fluoresce red from the mCherry. If the reporter was alone, it would only fluoresce blue from the CFP. If there was unmutated UL56 and the reporter, there would still be red and a little bit less blue than the reporter alone. The results of the unmutated UL56 plus reporter would be compared with those of the mutated UL56 E237D. Both would have the same levels of red. However, if the mutated has more blue, then the E237D has a lower affinity for the A site than the unmutated. If it has less blue, then the E237D has higher affinity for the A site. If it has the same, then the mutation has no effect.